So we will come to the next topic, that is a very important question for the exam point of view also, and we learned something called as the final pathway where we are going to synthesize the ATPs. So which is that pathway? That is the pathway which is called as electron transport chain. As the name itself tells, the information of this pathway where there is some electrons being transferred, and is this transfer of electron random? No. The transport of the electron or transfer of the electron is in a chain manner, means there is something which is present in a complex, it is present in a linear fashion and they are connected to each other, and through them the electrons are being transported. With the transport of electrons, what we are actually doing? So that is what we will be learning, and how it is transferring, that we will be seeing, and how this electron transport chain generates the energy, as well as this energy utilized in two purposes. One is ATP generation, other is the heat generation. How? We all know ATP generation from the electron transport chain, yes, we know it. But can electron transport chain generate the heat? Yes, it do generate the heat. So that also we will be seeing in this topic, that is how the electron transport chain helps in the heat generation, that is the generation of heat. So coming to the main topic, when we see the electron transport chain, what is happening is the transfer of electrons from reduced coenzyme A in the respiratory chain to oxygen, which is known as biological oxidation. As it is very clearly mentioning, what is happening is transfer of electrons. So what is happening is a compound is getting oxidized and other compound is getting reduced. So these are kind of oxidoreductase or oxidation and reduction reactions occurring parallelly. Okay, one compound is oxidized, other compound is reduced. So by the transport of the electron, and the energy released during this process of electron transport, we are generating what? ATP. So this is called as coupling of oxidation with the phosphorylation. We are coupling two reactions, oxidation reaction coupled with the phosphorylation. This is also called as oxidative phosphorylation. Okay, there are other type of phosphorylation which you learn otherwise called as substrate level phosphorylation, where ATPs are generated other than the electron transport chain. Okay, at the substrate reaction itself, the heat generated so much that it can lead to the binding of ADP plus Pi to form a ATP, substrate level phosphorylation. Here we will be seeing about the oxidative phosphorylation, the oxidation reaction, that is the transport of the electron chain with the generation of energy which is sufficient to synthesize the ATP, that is the oxidative phosphorylation. And the electrons flow occurs from successive dehydrogenase enzyme together known as electron transport chain. I told you oxidation is occurring one side and reduction is occurring on another side. Successively oxidation and reduction reactions are taking place so that the electron transport chain is happening. Now when we see this, we should know how they are organized, and secondly where they are organized. Now these are organized in mitochondria. Now we all know mitochondria has got a membranes, it has got the outer membrane and it has got the inner membrane. Okay, outer membrane and the inner membrane. So where is it electron transport chains are arranged? They are arranged in the inner membrane. Okay, they are arranged in the inner membrane, not the outer membrane. The electron transport chain, the enzymes or the complex or the multi-enzyme complex are present on the inner mitochondrial membrane, not the outer mitochondrial membrane. So all the components of the electron transport chain are located on the inner membrane of the mitochondria. And the electron transport chain or respiratory chain, what is happening is transfer of electrons, that is from the NADH to the chain of electron carrier. So it will carry the electrons. So electron travels from, very basic knowledge, that is more electronegative component or molecule to the more electropositive component, that is the flow, how it is done. Now if we see the electron transport chain, I told you it is a multi-enzyme complex. When I am telling a multi-enzyme complex means it contains multiple subunits or the domains. So multi-proteins which are present, they are termed as complexes. There are 4 complexes of the electron transport chain which are arranged in a fashion on the inner mitochondrial membrane, only on inner mitochondrial membrane. So complex 1, complex 2, complex 3 and complex 4. And there are 2 connecting mobile carriers. One is coenzyme Q and other is the cytochrome C. So total 4 complexes complex 1, complex 2, complex 3 and complex 4, and 2 mobile carriers which is called as coenzyme Q and also cytochrome C. So these are the 2 other mobile carriers. So it contains 4 complex and 2 enzymes. So when we see individually, the complex 1 contains few components, that is the FMN and then FES complex. Okay. Otherwise it is also called as NADH coenzyme Q reductase. NADH is getting oxidized and coenzyme Q is getting reduced. So I told you initially only the reaction is one is getting oxidized, other is getting the reduced by the transfer of electrons. So who is getting oxidized, who is getting reduced, we will see. Now NADH is oxidized into NAD plus with donation of H plus ions, which is reduction. FMN is reduced to FMNH2. Now the electron are transported to next FES complex, iron sulfur complex. So the oxidized form of FES accepts the hydrogen and forms a reduced complex. Further it will not keep quiet. What it will do, it will donate to the coenzyme Q to form QH2, reduced form of Q. So oxidized is converted into reduced. So you can see that the electrons are transported from FMN to coenzyme Q. Okay, so the transport of electron by oxidizing and reduction reaction. Next, now it will go to the second complex. Complex 2 is majorly made up of the iron sulfur complex. Here again same, the oxidation of succinate to fumarate to synthesize a reduced form of FAD, that is FADH2. The reduced FADH2 will get oxidized to the FAD with the donation of the hydrogen ions or electrons to reduce the iron complex. The reduced iron complex will donate the electron or H plus ion to the same coenzyme Q is converted into QH2. So it will accept the hydrogen ion. So one is oxidized, other is reduced, that is the electrons from FADH2 enters at this level and it is given to the coenzyme Q. Now this is about complex 2. Now third is the complex 3. So here this is cluster of iron sulfur proteins. It is cluster of iron sulfur proteins along with the cytochrome B and cytochrome C1. All these are what? They are heme containing proteins, means they contain the iron in them, which will be oxidized and reduced. How? Like QH2, coenzyme QH2, which is reduced, is getting oxidized with the donation of the electrons, where the iron is getting reduced. Fe plus 3 is converted into Fe plus 2. Further it will donate the electrons to the cytochrome B, where Fe plus 3 of the cytochrome B is converted into the Fe plus 2. So similarly further reaction goes to the cytochrome C and lastly to one more mobile carrier, cytochrome C, which will be from the oxidized state is converted into reduced state, means electron has transported till the cytochrome C, that is the mobile complex. Now last complex which is left is the complex 4. Now here complex 4 contains cytochrome A to A3. Okay, it is tightly bound to the inner mitochondrial membrane, cytochrome 4. Again same, the reduced compound donates the electron to convert the cytochrome A to 3 into a reduced form, and finally there should be someone who can accept this, that is done by oxygen. Oxygen accepts this with formation of a water molecule. So what happened at the end? The electrons are transported to the oxygen, molecular oxygen, to form H2O. Okay, at the end we are getting water out of this reaction. Is it only the electrons are being transported or something else is happening behind? So we will see that in the next also. So till now what happened? We have oxidation and reduction happening together with utilization of oxygen, and we are converting into water molecule. That is why the electron transport chain is also called as respiratory center of the cell, because when we tell respiration we utilize oxygen and we excrete the carbon dioxide. So utilization of oxygen is here. O2 is converted into water by accepting the electrons and finally reduced to water molecule. So when we see the whole complex, it is arranged where? It is arranged on the inner mitochondrial membrane, and there are 4 complex. I told you first complex that is FMN and FES, that is the iron sulfur complex. Complex 2 is iron sulfur complex. Complex 3 is FES and cytochrome B and cytochrome C1 complex, and then cytochrome A1 to A3. There is a fourth complex. Apart from this, I told you there are two mobile complexes, coenzyme Q and cytochrome C. So these are the two mobile complexes which are present. So when we see the reducing equivalents when they enter the electron transport chain, NADH2 and FADH2. So what happens when they donate the electrons to these complexes, and electrons are being carried from complex 1 to Q, there is coenzyme Q, from there to complex 3, complex 4. Not only electrons are being transported, they are also pumping the hydrogen from inner matrix into the intermembrane space. Okay, this is inner membrane. You can imagine here is the outer membrane. So between the space, there is the intermembrane space, we are pumping the hydrogen ions. So electrons are not just traveling, apart from just transported from one complex to other, the energy which is liberated during the transport of electron is utilized to pump the hydrogen in the inner membrane space. So when the electron transported from complex 1 to coenzyme Q, we pump 4 hydrogens. When electrons are transported from complex 3 to complex C, that is cytochrome C, we pump 2 hydrogens, and then last complex 4 to water molecule formation, we pump 4 hydrogen ions. So total 8 hydrogen ions per 1 electron transport through the whole cycle. At the end, what we are generating, half mole of oxygen is converted into water molecule. So we are generating the water molecule at the end with transport of the electrons and pumping the hydrogen into the intermembrane space. Now when we are pumping the hydrogen from matrix to the intermembrane space, what we are actually doing is we are generating a gradient of hydrogen. Normally the hydrogen concentration here is very high. The matrix contains very high concentration of the hydrogen, whereas the intermembrane space is very limited, and when we pump lot of hydrogen is there, what we are generating is a complex or a compression of hydrogen, that is we are generating a force. This force is for what use? That we will see in the next further. Okay, how it is related to the ATP synthesis we will understand with this hydrogen ions. So till now the electrons are transported, half session is done. What is that? Electron transport chain. What we have done? We have transported the electron. We have transported the electron from the reducing equivalent to the oxygen to get converted into a water molecule. Meantime, during the transport of electron, we generated some heat or energy. That energy we used to pump the hydrogen ions from the inner membrane space into the intermembrane space, or from the matrix to the intermembrane space. So that is about the electron transport chain. There is one more small concept comes as PO ratio, which can be asked as a 3-mark question in the exam. Now finally at the electron transport chain, what we are consuming is oxygen. We are consuming the oxygen. So the PO ratio is defined as the number of inorganic phosphates molecules which are incorporated into ATP, means how much oxygen used to synthesize ATP. So that is called as PO ratio. When a pair of electron from the NADH reduces an atom of oxygen, that is half O2, 2.5 moles of ATP is formed. 2.5 ATPs are formed. When it is NADH, when it is FADH, because it enters in the complex 2, the first 4 hydrogen ions are not pumped into the intermembrane space. So total hydrogen ion which are pumped are only 6. So hence it can add up to only 1.5. Okay, conversion of oxygen into water. When it is NADH2, it is synthesizing 2.5 ATP, and when it comes to the FADH2, it synthesizes only 1.5 ATP. So that is the PO ratio. Now till now we have seen only the transport of electron, how oxygen is converted into water, where the hydrogen are pumped. But still we have not touched where is this ATP generated. We are only seeing the other things but not the ATP. So now the ATP synthesis occurs at specific site. It is not these 4 complexes will generate. Initially what they thought is by pushing the hydrogen ions into the intermembrane space, will generate the ATP. But with the generation of the information and evidence, they understood that it is not pumping the hydrogen into intermembrane space which is generating ATP, but it is something else. Okay, so traditionally the site of ATP synthesis were marked as site 1, 2 and 3, which we have seen in the electron transport chain. Okay, so next coming to the concept where they claim or theory where they explain how this transport of electron, how the formation of hydrogen gradient is contributing to the ATP generation. Okay, that is termed as oxidative phosphorylation, that is oxidation with phosphorylation coming together to make one process, oxidative phosphorylation, that is transport of protons from the matrix into the intermembrane space accompanied with generation of one important gradient, that is the proton gradient across the membrane. It's like a pressure cooker, you are adding a pressure in the intermembrane space. So this accumulate outside the membrane, creating a electropotential difference between what? Intermembrane space and the matrix. So there is generation of a gradient, and how you are using this gradient will define the ATP generation. That is, this proton motive force derives the synthesis of ATP. Who will synthesize? There is specific complex called as ATP synthase complex. There is a fifth, we also term it as a fifth complex of the electron transport chain, which is the ATP synthase complex. So ATP synthase complex is the one who can only generate the ATPs, okay, from this gradient. So in exam, for 3 mark question they will ask you to write about ATP synthase. So you should be able to draw the diagram also. Now again, when we see this diagram, we understand it has some parts. It has made up of different proteins which are subunits. So it is a protein assembly which is present where? Again it is the inner membrane, not the outer membrane, inner membrane, and it is hanging into the matrix of the mitochondria. Okay, and it is also termed as the fifth complex, and it is differentiated into 2 fragments or segments, that is the F0 and F1, okay, FO and F1 complex. FO is submerged in the membrane, okay, it is part of the membrane, and the remaining portion, that is the F1, is in the matrix, okay, it is hanging in the matrix. The F1 complex is hanging in the matrix, and FO will act like a hanger, it will be embedded within the membrane. So this has 2 functional, which already I told you. Then when we talk about the mechanism, how it will generate, now this is fine, we have understood that it is structure containing of O and F1 complex, but how they are catalyzing. So translocation of proton carried out through the FO complex, formation of a bond between the ADP plus Pi. Okay, so the binding change mechanism, which is proposed by the Paul Boyer, who got the Nobel Prize for the same, explains the synthesis of ATP from this complex, that is the ATP synthase complex utilizing gradient. How? The FO complex acts like a wheel, okay, it will act like a wheel which can get rotated, and when the proton gradient moves through this, okay, if we imagine this is a wheel, when the proton gradient move towards this from the intermembrane space to the matrix, what it will do? It will blow with the force, it will blow with the direction. So this wheel starts rotating, there is rotation of the FO complex. When FO complex rotates, what happens to the F1 which is hanging? Definitely it will also rotate, which contains gamma, alpha and beta portions. So it will also rotate. So this rotation, like you might have seen turbine, when the turbine rotates you will generate the electricity. Same way here, when the turbine rotates, now windmill rotates because of wind force. Now here this ATP synthase will rotate with the help of H plus gradient, the hydrogen gradient which is generated. So when this is happening, according to this theory, rotation you cannot explain the synthesis of ATP. What they told that with the help of rotation there is some conformational changes, because again these are what? Proteins. So proteins will have their conformational changes, they will have their own structural catalytic activity. So they have 3 forms. What forms? 3 forms: O form, L form and T form. What they stands for means whether ADP and ATP are binding to this complex or not. Okay, when we see it contains 3 forms, O form, then we have got L form and T form. O means no, it has got no affinity for the substrate. Who is the substrate? It can be ATP or ADP. No affinity means that is the place, like for example if you see the 3 places, okay, you have got O, you have got T and you have got L. So when we see 3 forms, O form does not have any affinity for its substrate. When it comes to L form, it has little affinity to the substrates. It can be ADP, so it can bind to the ADP plus PI. When it comes to T for tight means what it is doing, ADP plus PI, it is combining. To combine what? ATP. By tight junction we are generating the energy. So by rotating the whole complex, what we are doing is 3 conformational changes. Like for example, you are making some laddu. What you will do? First you will hold lightly, then what you will do? You will press it last tight, and then when you leave it, you have got your own product. So what is the product here? ATP. So we are generating the ATP by this conformation O, L and T. O means no affinity, L means little affinity, T means tight affinity. It will bind very tightly. Okay, so when we see that mechanism also, first what happens? To generate ATP, what should be available? We want ADP, we want phosphate groups PI. So they both are available. So where they are binding? They are binding on L. What is binding? ADP plus PI. They bind at the L position, binding site. Now when the wheel rotates, when the ATP synthase rotates, this will take this position. Imagine. So it will go to T conformation. What happens? Because of the tightening, we generate ATP. So that is, we are formation of ATP. But once it is formed, do you want it to sit there only, or we want it to be released in the matrix? We want it to be released because the work is not with the ATP synthase. The function is done. We have synthesized the ATP. Now it has to go for its function. So now it undergoes third change. What is that? The conformational change is O. What happens? It will release the ATP. No affinity for the substrate. It can be substrate, can be ATP or ADP. No affinity at all. So it will release. Now again this cycle repeats. L form binds with one more ADP plus PI, then it will rotate to T form. T form will combine them both and form an ATP. O form will release it, then again it rotates. So during the rotation, ATP is generated. Who is rotating it? The blow from the hydrogen ions. The hydrogen ions are gradient. They are making this turbine to rotate. When the turbine is rotating, it is making some conformational changes, like it is holding tightly and again releasing it back. So the conformational changes to form the ATP. So that is how ATP is generated. That is the mechanism of ATP generation. Now last one, when synthesis is occurring, definitely we require someone to regulate it. Who is regulating? First, availability of the ADP. When there is lot of ADP, availability of lot of ADP means very less ATP. So we want electron transport chain to generate lot of ATP so that it can be used for energy purpose. Next, when ATP levels are low and ADP levels are high, so again also the electron transport chain will be at the rapid. So phosphorylation proceeds at the rapid rate, and this is called as respiratory control or acceptor control. Okay, at the center only you are deciding how it should run. And next major is the reducing equivalents. If there are no reducing equivalents, no starting point for the electron transport chain, the chain itself will not act. So the starting point is what? The reducing equivalents, availability of NADH and FADH2. If they are present in lot, electron transport chain runs at fast. If there is deficiency, then again electron transport chain also will be running at very low, which will be causing the energy loss in the cells. So this is how these are regulated in the body. Now last section comes is most important question, that is inhibitors. This is the important 3-mark question as well as 5-mark question in the exam. Okay, they will ask you write the inhibitors of electron transport chain. Sometimes for 5 marks they will ask you to write specifically complex wise any 3 complex and inhibitors, or sometimes 3-mark question they'll ask you, write any inhibitors of electron transport chain. So we should know complex wise. Easy to remember. Complex 1 inhibitor, we have got the barbiturates, fish poison, insecticides, chlorpromazine and some antibiotics. So these are the most common drugs which are taken, like barbiturates, anti-seizure, antibiotics are taken, insecticides through contamination of fish poison, also through contamination. So these all will inhibit the electron transport chain at what position? At the complex 1, they will inhibit the electron transport, they will stop the ETC. Next, complex 2, who can inhibit? Carboxyl. So carboxyl is a molecule which can inhibit the complex 2. Next, complex 1 and complex 2 are done. Now who can inhibit complex 3? That is the BAL, naphthoquinone and also the antimycin. So these are the 3 important drugs and poison which can inhibit the complex 3. Okay. Next, complex 4. Now most commonly in the exam they will ask you, family was living in a room and they were using the heater which was not maintained properly, and morning they were all found dead. What is the cause? It is related to the electron transport chain. How? Because combustion, that is the partial combustion, will eliminate or synthesize lot of this molecule, that is the carbon monoxide. So carbon monoxide will inhibit the electron transport chain at the complex 4. So when electron transport chain is stopped, I told you, no energy, no cell survival, cell will die, individual will also die. So if the carbon monoxide is excess, or simple, you might have seen in the movies, people will take something from the mouth, immediately they will die. Cyanide poisoning, it will inhibit the complex 4 very efficiently. So cyanide poisoning, others includes like hydrogen sulfide or azide. So these are the other molecules which can inhibit the complex 4 and hence the electron transport chain is halted. Other than this, site at succinate dehydrogenase and complex Q, that is the second complex. Inhibitors are very less, one is carboxyl, other is malonate. Next, some molecules will inhibit only oxidative phosphorylation. They uncouple the reaction, which includes actocloside, oligomycin and valinomycin. Again they are the antibiotics, they can inhibit the reaction, that is the oxidative phosphorylation. Apart from these all things, further one more important question what we might expect in the exam most commonly are the uncouplers. We can ask for 3-mark question or even for 5-mark question explaining various mechanisms. So we will see that. So what are the uncouplers? I told you oxidative phosphorylation is a coupled reaction where oxidation parallelly, in other hand phosphorylation. Oxidation with phosphorylation, the whole electron transport chain is helping to synthesize the ATP. Now what we are doing? We are uncoupling. Oxidation is happening but phosphorylation is not taking place. So there is an uncoupling between these two mechanisms. So they are called as uncouplers. There are some chemical uncouplers and also physiological uncouplers, means they are already present in our body who can uncouple these kind of reactions. So first we will see what are the uncouplers which are chemicals, that is 2,4-DNP, that is 2,4-dinitrophenol. So that is a one important uncoupler. One more is 2,4-dinitrocresol is another molecule which can become a uncoupler, and other is CCCP, that is the chlorocarbonyl cyanide and then phenyl hydrazine. So which is an important uncoupler. These are not normally present. They are the chemical substances, when you take them or inhale them or contaminate your food, definitely you are going to have the uncoupling. But in our body there are already presence of some couplers, uncouplers. They are called as physiological uncouplers. In exam for 3-mark question they will ask you, write the examples for physiological uncouplers. They are important. One is the thyroid hormone. Thyroxine itself is a important uncoupler in high concentration. Next, one more is called as thermogenin. Thermogenin is an important molecule which is present in the adipocytes or adipose tissue, brown adipose tissue, which also uncouples this, means it is separating the both. Oxidation is occurring but it is not coupled with the phosphorylation or ATP generation. So they are called as uncouplers, physiological uncouplers and other uncouplers. Now next we will see what are the importance of these uncouplers. Are they important? Now if you think we are telling this is physiologically present, when we are telling it is physiologically present, definitely it is of some importance. There is some what you call the importance of this uncoupling in the body. What is that importance? If you separate them, somewhere the energy has to dissipate. The proton gradient, if not through ATP synthase complex, it will enter the matrix, but whatever the energy is there, it is dissipated. Where is it dissipated? It is dissipated as heat. Okay, the energy is dissipated as the heat. So uncouplers will allow oxidation to proceed, but energy, instead of trapped through phosphorylation, is dissipated through the heat. Like for example, you have got the intermembrane space. This is the ATP synthase complex. Instead of hydrogen coming through this complex, what you will do, you will make one more pore here, so that all the hydrogens are coming through this. So the energy is same, energy is present. Instead of rotating this turbine, what it generates is, it generates the heat. Okay, so this is the mechanism how the body generates the heat. The gradient is same, the hydrogen gradient, instead of ATP synthesis, we are utilizing it for the generation of heat, that is the removal of proton gradient through the other channel other than the ATP synthase complex. So uncoupling is useful biologically because we are generating heat, and then in the specific situations like hibernation in most of the animals, so in the children's, that is human infants, the heat generated, because in human infants, now we all know that adult generate heat by the muscular activity. When it comes to children, the muscles are very hypotonic and inefficient to produce heat. But who has to take care of this heat generation? The body has to maintain the normal temperature. So that is through the heat generation through the adipose tissue. Adipose tissue is rich in a molecule called as thermogenin. Thermogenin is a physiological uncoupler. So because of which the heat is dissipated, the child will end up generating the heat, that is through the brown adipose tissue. That's why we tell that newborns and infants are containing rich number of adipose tissue. Which adipose tissue? Brown adipose tissue. What we have? White adipose tissue. White adipose tissue is named because it contains lot of fatty acids. When it goes to the children or newborns, infants, actually it is majorly containing lot of mitochondria. It gives the brownish tinge to the cell or the adipocytes. So it's called a brown adipose tissue. So it dissipates the heat. That is thermogenin, a protein present in the mitochondrial membrane of the adipocytes, provides an alternative route through which protons can be entering the matrix. Hence it will generate the heat, and it is one of the important uncoupling protein, thermogenin. And one more is thyroxine hormone. If it is in high concentration, then also the body temperature is increased. Why? Because it also acts as an important physiological uncoupler of the whole reaction. So by this what we understood, that electron transport chain is not only synthesizing ATP, but also it is important for maintenance of most important compound, that is the generation of heat, so that it maintains a normal body temperature. Okay, so with that we will be concluding on the topic. Before we wind up, we have to summarize some important questions how we are going to do. One is the electron transport chain itself is the important major question. They will ask you to write about the electron transport chain complex, draw the neat level diagram and explain how ATP is generated, or they will ask you to write about chemiosmotic theory or theory by which ATPs are generated. Next, they might add one more point in that, write some inhibitors of the electron transport chain. So you should be able to write complex wise, what are the inhibitors of complex 1 to complex 4, and then also uncouplers. Then write specifically 5 mark, they might ask you 5 mark question, they might ask to write about the inhibitors of electron transport chain. What are the inhibitors at different complex? Or last, they might also ask, draw a neat level diagram of electron transport chain. This again can be a 3 mark question, this can be a 10 mark question, or sometimes they will ask you to write 3 mark question like write a note on brown adipose tissue, or write a note on how uncoupling will help in the body temperature regulation, or write a note on thermogenin. So these are the other few questions which can be expected out of this topic, that is the electron transport chain, inhibitors, uncoupling, how the gradient is used for oxidation and phosphorylation. So with that we will be concluding on topic electron transport chain. Now we will be coming to another topic which we will be discussing, that is called as ketone body metabolism. Now when we come to this point, does ketone body come under which metabolism is the question. So when we see that, it comes under the lipid metabolism, because ketone bodies are the derivatives of the same, and ketone body metabolism is very important in two situations. One situation is uncontrolled diabetes mellitus, and second situation is when the person is starving or he is not on the carbohydrate diet for a very long time. So under these two situations, this topic comes into a major importance, that is the ketone body metabolism. So when we go ahead, we will be seeing that a statement called as fat is burnt under the fire of carbohydrate. Like you have seen a deep where the lamp is burnt with the help of the oil. So similarly in the body also, fatty acids or the lipids are burnt if there is presence of carbohydrate. If there is no carbohydrate, then this lipid metabolism also will be hampered. So that is the first statement. Second, as I told, carbohydrate plays a very important role for the fat metabolism. Thirdly, the acetyl-CoA, which is formed from the fatty acid degradation, that is the beta oxidation, enters the TCA cycle to get completely oxidized in the presence of carbohydrate. But during the starvation and uncontrolled diabetes mellitus, what happens is this acetyl-CoA, the acetyl-CoA which is derived from the fatty acid degradation, will take alternative route. Why? Because the TCA cycle is busy in synthesizing glucose. The glucose or gluconeogenesis pathway, the intermediates are taken from the TCA cycle, because of which what happens, TCA runs at a very slow rate. During this slow rate, there is no sufficient speed to catch up with the oxidation of acetyl-CoA. There is accumulation of acetyl-CoA. The acetyl-CoA start getting accumulated. The acetyl-CoA accumulation will lead to its alternative pathway. The acetyl-CoA, what is getting hampered or what is getting accumulated in the mitochondria, takes alternative. That alternative is the ketone body synthesis. Now in exam, they will ask you to write what are the examples for the same. You can expect 3 mark question in that. So you should be able to write 3 major important ketone bodies, that is acetoacetate, which is also called as primary ketone body, because during the formation of ketone body, it is the first ketone body which is synthesized. So that is why it is called as primary ketone body, and next remaining 2 are called as secondary ketone bodies, because during the synthesis they are synthesized second. Okay, so they are synthesized second, they are called as secondary ketone bodies. Now next, when we see the synthesis, that is the ketogenesis. This is an important exam question which is a 5 mark question in the exam. They will ask you to write the pathway how ketone bodies are synthesized. Before we go that, we should always know 2 things. What is the site? Site is majorly the liver. Liver is the only organ who is capable of synthesizing ketone bodies. In the liver, what is the site? That is the mitochondria, because the substrate which is required for the synthesis, that is the acetyl coenzyme A. Acetyl coenzyme A is present in the mitochondria, and beta oxidation is also occurring in the mitochondria. Hence, the substrate which is required for ketone body synthesis is also present in the mitochondria. From the mitochondria, it takes diversion towards the ketone body. As I told already, TCA is running slow. There is accumulation of the substrate, that is acetyl CoA. Now, acetyl CoA has to undergo some metabolic pathway to supply the energy to the body. So, it takes the alternative, that is the ketone body synthesis. How it happens? It is a simple step of calculation. 2 plus 2, 4, which is acetyl CoA. That is how many? 2 acetyl coenzyme A condenses with each other with the help of the enzyme thiolase to synthesize acetyl coenzyme A, which is a 4 carbon compound. From 2 carbon compound, it is converted into 4 carbon compound, which is acetyl coenzyme A. Now further, acetyl coenzyme A undergoes one more addition of 2 carbon compound, that is acetyl CoA, to synthesize HMG CoA, that is beta hydroxy beta methyl gluteryl coenzyme A, HMG coenzyme A in the mitochondria. Now the HMG CoA will release the acetyl CoA. It will lyse. HMG CoA is lysed by HMG CoA lyase to synthesize the first ketone body, that is the primary ketone body. As I told you, acetoacetate is called as the primary ketone body because during the whole pathway, that is the first one to be synthesized. Now this acetoacetate will have 2 fate to synthesize 2 successive ketone bodies, that is secondary ketone bodies. One, it will undergo reaction, that is the hydroxybutyrate dehydrogenase, to synthesize beta hydroxybutyrate, which is a more of water soluble compound, or it will undergo secondary ketone body synthesis, that is the acetone, that is the synthesis of acetone by decarboxylation reaction. You are removing the carbon dioxide from the acetoacetate to synthesize acetone, and acetone, as you have already come across, it is very much volatile. Okay, with the reaction, that is acetoacetate decarboxylase, by decarboxylation reaction carbon dioxide is released and we get acetone. So these are the primary ketone body, that is the acetoacetate. Secondary ketone body, we have got the acetone and beta hydroxybutyrate. These are the two secondary ketone bodies in the ketogenesis or ketone body synthesis, occurring where? It is occurring in the liver. In the liver, what is the site? Specifically subcellular organelle, it is the mitochondria who is helping. What is the substrate? Substrate is the acetyl coenzyme A. What is the end result? We are getting the ketone bodies. 3 ketone bodies, we name it as acetoacetate, primary ketone body, and 2 secondary, that is the beta hydroxybutyrate and acetone. Out of beta hydroxybutyrate and acetone, acetone is volatile, beta hydroxybutyrate is water soluble and it is excreted through the urine. Okay, now next comes is regulation. How they are regulated? Normally they are regulated at 3 crucial levels. One, availability of the acetyl CoA. Now availability of the acetyl CoA depends on one oxidation process. What is that? Beta oxidation. That is how much free fatty acids are available from the adipose tissue for beta oxidation. More beta oxidation, more acetyl CoA will lead to excessive ketone body generation if TCA is hampered. Okay, so availability of the free fatty acid. Next, one important molecule required for transporting palmitic acid or the long chain fatty acid into the mitochondria is carnitine shuttle. If carnitine shuttle is properly working, then it will, if you have inhibition of the carnitine shuttle, then again ketone body generation will be low. Then partition of acetyl CoA between the pathway of ketogenesis and the citric acid cycle. If citric acid cycle is running very fast, then ketone body generation will be at very minimal. If TCA cycle is running at the minimal, then ketone body generation will be at the fast, because the same substrate will enter 2 doors. If one door is closed, other is always open and it is flooded. So same way, if TCA is running at very low rate, where you see in the case of starvation, or you might see in the case of the uncontrolled diabetes mellitus, where gluconeogenesis pathway is taking on, the oxaloacetate and the other intermediates on the TCA cycle are taken up. So TCA runs at a very slow rate because its intermediate are not refilled back. So it is running at a slow rate. These are the 3 crucial levels at which ketogenesis is regulated. Next, we have synthesized it. Do we have to utilize it, or do we have a mechanism to utilize the same ketone bodies? Yes, that is comes through the ketolysis, that is lysing the ketone bodies. Now here one important thing is, the organ which synthesizes cannot utilize it. Okay, that is the liver is the organ who will synthesize the ketone bodies, but ketone body utilization is through the other organs. We call it the extrahepatic utilization of the ketone bodies. So tissues other than the liver, that is extrahepatic tissues such as, which includes the skeletal muscle, heart muscle, or your renal cortex, especially the brain tissue when glucose is low. So these are the organs who will utilize the ketone body for their survival when the glucose levels are very minimal. That you see under uncontrolled diabetes mellitus, where sugar is high, the glucose is high, but it is not entering into the tissue or the cells. Or second, in starvation, where glucose itself is very low in the person. So those 2 conditions, these extrahepatic tissues start functioning or start utilizing the ketone body. At subcellular level, what is the site? It is again same, mitochondria. So all the organs or the tissue which has got the mitochondria other than liver can utilize the ketone bodies by a process called as ketolysis. It is almost similar to reversal. Okay, because we want finally what? Acetyl CoA or acetyl coenzyme A, because that has to enter the TCA cycle. In the TCA cycle, it has to undergo the one cycle and synthesize the ATP which is required as energy purpose. So definitely it is almost similar to reversal. Like for example, acetone, because it is volatile in nature, it is excreted through the lungs. So we might not have the acetone sufficient to act as the energy supply. What we left behind is a water soluble compound, that is beta hydroxybutyrate. So beta hydroxybutyrate will undergo dehydrogenase to synthesize the acetoacetate. Okay, acetoacetate we got, it's a 4 carbon compound. Acetoacetate will undergo transferase reaction. Okay, it will undergo transferase reaction with the help of succinyl coenzyme A. What we actually require is one coenzyme A. So that coenzyme A is donated by the succinyl CoA. So succinyl CoA reacts with the acetoacetate by beta keto acyl coenzyme A transferase. What we transferred is coenzyme A. So acetoacetate is converted into acetoacetyl coenzyme A. Now acetoacetyl coenzyme A can be split into 2 because it is a 4 carbon. We can convert this 4 carbon into 2 different acetyl coenzyme A. What is required here now is we require one more coenzyme A. So one more coenzyme A is added by the enzyme thiolase, converting acetoacetyl coenzyme A and splitting into 2 compounds acetyl CoA. How many? 2. We get 2 acetyl CoAs from single hydroxybutyrate which enters the TCA cycle. Now please remember, whole this pathway is occurring in the mitochondria, TCA is also occurring in the mitochondria. So the substrates required for the TCA cycle are delivered at the same place, that is the extrahepatic tissue. And as acetone is excreted through the lungs, that is why I told you first only, it is not available for the ketolysis purpose. Okay, so this is the simple pathway how the ketolysis occurs in the extrahepatic tissue generating the energy. Okay, next one more term called as ketosis. This is an important 3-mark question in the exam. Okay, they can ask you write about ketosis. Ketosis means it's a condition where you have lot of ketone body beyond the capacity of your extrahepatic tissue to utilize it. So what happens finally? You are accumulating it. As I told, the ketone bodies are beyond the capacity of the extrahepatic tissue to oxidize, utilize it, eat it. So what happens? You have lot of ketone bodies in your blood. So there is increased levels of ketone bodies in the blood, that is called as ketonemia. Ketonemia means in the blood the ketone body concentration is increased. Now because it is water soluble, what happens? Will it be excreted in the urine? Yes, definitely. You will find excretion of these ketone bodies in the urine, which is called as ketonuria. And overall ketonemia plus ketonuria, the condition is called as ketosis. In the exam they will ask you for 3-mark question, what is ketosis? So ketosis is a condition where there is 2 things. One is ketonemia, that is increase in the blood levels of the ketone body beyond its normal level, and because it is high in the blood, water soluble starts excreting the urine, which is called as ketonuria. So hence we term the condition as ketosis. So that is an important exam question for a 3-mark, ketosis. And last, where do you see this? Okay, ketone body high ketonemia or ketosis, you will see in 2 major conditions. One is the starvation, because we do not have the metabolites for the TCA cycle because we are at the high rate of gluconeogenesis. Second, uncontrolled diabetes mellitus, not the controlled, uncontrolled diabetes mellitus, because again here there is excessive amount of glucose in the blood, but there is no supply to the cell. What cell will think? That there is no glucose, because nothing is coming in, definitely it will think that there is no glucose available. So these are the 2 conditions where it will trigger for fatty acid degradation, acetyl CoA synthesis excessively, all the intermediates of the TCA are utilized for the gluconeogenesis, hence acetyl CoA starts accumulating, they end up into the alternative fuel for the body, that is ketone body. Other thing includes high fat feeding and also after severe exercise in post absorptive state, because again severe exercise definitely the ingested glucose will be vanishing, the stored glycogen will be vanishing, gluconeogenesis will taking care of the glucose levels. So in such situation again the acetyl CoA starts accumulating and it will lead to ketosis. So acetoacetic acid or acetate, beta hydroxybutyrate are buffered by bicarbonates. So whenever there is increased levels in ketone bodies, what happens? You are utilizing the alkali, that is the bicarbonate, for neutralizing these acids. So when you are neutralizing these acids, the person will end up into a acidosis because alkali reservoir are reduced. So person will end up having the ketoacidosis, that is acidotic condition due to decrease in alkaline reservoir. For what reason? Due to excessive ketone bodies. The more amount of ketone bodies we utilize the bicarbonate buffer, decreasing the reservoir. Decreasing reservoir of bicarbonate will lead to the acidosis, and acidosis is due to ketone bodies, hence we call it as ketoacidosis, which is majorly seen in one condition, that is uncontrolled diabetes mellitus. In uncontrolled diabetes mellitus or in type 1 with low insulin or no insulin levels, the person or child will end up having this DKA, diabetic ketoacidosis, where ketoacidosis is because of diabetes mellitus. Okay, it can be type 1 uncontrolled or even type 2 uncontrolled. So, this is about the ketone bodies. And lastly, to summarize, we have got the tissue where the ketone bodies are generated. I told you, liver. How it is synthesizing? It is synthesizing from the acetyl CoA. From where we are getting the acetyl CoA? We are getting from the beta oxidation. Beta oxidation of what? A fatty acid or acyl coenzyme. What is the location? Mitochondria is the location. Under conditions where the TCA is running at very slow, the acetyl CoA cannot enter the TCA cycle, so it takes the alternative pathway. What is the alternative pathway? Is ketone body synthesis, the primary and secondary ketone bodies, that is the acetoacetate, beta hydroxybutyrate and acetone. Now, when they are entering into the bloodstream, acetone, because it is a volatile in nature, it can be excreted through the lungs. Now, what is left behind is the beta hydroxybutyrate and acetoacetate. So, these two at the tissue level. What tissue level? Extrahepatic tissue, because I told you already, extrahepatic tissue is the one which can utilize the ketone bodies. So, extrahepatic tissue almost similar to the reversal of the ketogenesis. So, ketolysis occurs, we generate acetyl CoA, from one ketone body we get two acetyl CoA, those two will enter the TCA cycle and generate the ATP, which is required at the place. Okay, so extrahepatic tissue utilizing the ketone body, which is called as ketolysis, liver generating the ketone body, which is called as ketogenesis, acetone, because it is volatile in nature, excreted through the lungs. So, these are the few important things you should know, how the ketone bodies are metabolized in total in the body. Okay. So, lastly, how you differentiate two things. I told you two commonest condition for ketone body generation, which is ketosis. One is uncontrolled diabetes mellitus, that is DKA, diabetic ketoacidosis. Second is the starvation. So, when you do the blood and urine investigation of the patient, like for example in diabetic ketoacidosis is condition where both are increased. There is already high level of glucose which is excreted in the urine. Ketone bodies are also very high because cells senses it as no glucose or low glucose level. So, when you do the urine testing for the glucose, Benedict test, it will be positive in DKA, and Rothra's test for the ketone body, it is also positive. So, both glucose and ketone body are positive in the DKA or diabetic ketoacidosis. When you see the patient with starvation, urine test for Rothra, that is ketone bodies, will be definitively positive, but because person is in starvation, already is in hypoglycemic state, there is less amount of glucose. So, do you think it will excrete in the urine? No. So, you will see negative for Benedict test. So, whenever the patient comes with ketosis, when you do urine testing for glucose and urine test for ketone, Rothra's test and Benedict's test, Benedict test and Rothra's test are positive in DKA. When it comes to the starvation, the glucose is very low, so definitively it is not excreted in the urine, so it is negative for the urine, Benedict's test, and Rothra's test is positive for the ketone body. So, this is how you are going to differentiate the type or cause for the excessive ketone bodies in the blood. Okay. And lastly, what is the take home? What are the important questions you might expect in the exam, so that you will be able to answer it? One, briefly describe the mechanism how ketone bodies are generated. So, they are asking you ketogenesis. So, you should write what is the reason, what is the mechanism, where it occurs, what is the site, liver, within the liver where is the site, mitochondria, then what is the substrate required, what is the end product synthesized, then steps you have to put on the flow, and then write about some points on ketosis. Next, second question, which is most commonly asked for 3-mark question or short answer question, name some ketone bodies or name any 3 ketone bodies. So, you have to differentiate both, do not write randomly all 3, write primary ketone body acetoacetate, secondary ketone body beta hydroxybutyrate and acetone specifically. Then organ synthesizing and organ utilizing the ketone bodies. Again, it is a short answer question. What is asking is which organ utilizes the ketone bodies. So, that is the extrahepatic tissue, name them. Then which organ synthesizes the ketone body, which is the only liver. Liver can generate the ketone body. So these are the three important questions you can expect in the exam, and also you should be able to give reason, like in the last slide we have seen how the ketone bodies are different in diabetic ketoacidosis and how you are going to differentiate the ketone bodies excretion in the starvation, two conditions for ketoacidosis. So with that we will be concluding for the topic ketone body metabolism. Thank you.